Thank you very much, Tom, for agreeing to give a Build a Cell seminar, and you can get started.
Cool. Yeah, thank you. So this is just a title slide. So yeah, firstly, thank you for the invite. I already said that, but officially with the recording, thank you. Very happy to be here. And because I'm supposed to do a kind of self-introduction, I thought I would just do a slide to make it easier and less awkward. So for those of you who don't know me, previously I was a group leader at the Max Planck Institute of Colloids and Interfaces in Germany. And since 1 year, I've moved my lab to the University of Edinburgh in Scotland. So, and there I'm a lecturer in chemical engineering. And my labs are physically based in the Institute of Bioengineering. So yeah, I'm also going to advertise if you're interested in doing a PhD after all of this, then please drop me an email. So yeah, so today I'll be talking about bottom-up engineering of synthetic cells, multiple compartments, and complex membranes. And for those of you who also don't know kind of what my lab does, this is an overview of what we do. So we use tools such as microfluidic technology, and I'll talk more about that later. And we combine them with processes and materials such as molecular self-assembly and soft matter materials in 4 main research areas. So the first one is single cell analysis, cell membrane biophysics, cell membrane fusion. And the last one, which is the main one, is bottom-up synthetic biology. And hence that will be the focus of this talk. So what my lab is interested in is looking at biological complexity and asking if we can simplify it, but also retain some of the functionalities. So what exactly do I mean by kind of complexities? Well, obviously we can take a microscope image of a living cell and we can see lots of structural complexities. Then we can infer, exactly what is going on. And this is kind of one of the pictures that we have of cellular complexity. But we also know that there's lots of biochemical complexity as well, which we cannot see directly. But these two things work hand in hand to bring about a diverse range of cellular functions. And the overall question in my lab is always, is this cellular complexity that we see today in cells really essential to bring about these functions? Or can other routes possibly exist to bring about similar functions? And what do I mean by cellular functions? Well, I can basically kind of break this down into, so I'm going to minimize that because I can't see myself, but hold on. There we go. You can break this down into different categories, and you can argue what are the most important functions, but here's just a few of them, ranging from compartmentalization to protein expression to cellular growth or division, etc. And the first research question I want to ask is, can we achieve similar functionalities but with simplified systems? And this brings us to the idea of what a minimal cell is. The second question is, can we use such bio-inspired systems to our advantage? So, can we find new applications in biotechnology or biomedicine, for example? So this is just one slide on what minimal cells are. So for those of you who don't know, a minimal cell kind of definition is the minimum number of components required for a cell to function. And the way that traditionally this has been kind of attempted or achieved is to take the top-down approach of synthetic biology, where you take an existing organism and you reduce its genome down to a minimum set of genes or proteins. And we all are kind of aware of the success of the Craig Venter Institute in achieving, lots of gains in this top-down approach. But there's still lots of genes which kind of their function's not known, so there's still a lot of open questions about how these things work. So the other approach, the alternative approach, is what my lab does, is to take the bottom-up one, where we construct a minimal cell from non-living components. And of course, you're right to be skeptical and say, you might not get to the same level of achievement as top-down approach, and that's true. But what we try and do is, along the way, we try and identify and mimic, individual processes and step-by-step things that we think are essential for life. And if we can get even partially there, then, we can understand the system that we build arguably better than the top-down approach. But like I said, it's basically the journey, not the destination. So minimal cells in general can be useful because they can help us understand complex biology. So we're interested in building them as cell models. And of course, they can have implications for origin of life because they might be able to tell us where we have this transition from matter to life. So the idea is that we take these non-living components and we also develop new enabling technologies, which we can also advertise well and publish. And we apply them to build up, using the bottom-up approach, our synthetic cells. And we do this, step by step, and eventually the long-term goal is to get this minimal cell that can do things like self-replication, proliferation, etc. And along the way, we hope to find new applications in biotechnology and biomedicine. So this is the vision. Now, whatever we try and do to build our synthetic cells, whether we take new technologies and develop new technologies, whether we take existing elements of biology or develop new chemistries, one thing is quite obvious, at least my argument would be that we need some kind of compartment-based system. So, and the requirements of that system would be that we need to encapsulate and concentrate the necessary molecules or ingredients. So what kind of materials could we use? Well, we could use coacervates, for example. We could use polymersomes to create polymersomes, proteins to create proteinosomes. We could also have droplets, so water and oil droplets or emulsions, and we could have liposomes or lipid vesicles. But even if we take those materials, there's also a variety of different structural assemblies that we can think about. So, we could combine. Can you still hear me? It said my internet connection's slow. Okay, fine.
Yes, we can hear
Assume you can
you.
still hear me. Okay, good. Okay, I had a warning that something was slow. If I cut out, tell me. So we could have combinations of different materials. So, for example, lipids and polymers. We could have networks of different materials. So we could have multisome networks, for example. And we could have two-phase aqueous systems. And the last option, at least in these choices, is that we could have vesicles nested inside other vesicles. And this is the choice of my lab. And more specifically, the material that we choose is lipids, so lipid membranes. And why do we choose them? Is because we think they're the gold standard, as they're highly biocompatible. I mean, they're already used in vaccines for drug delivery. In this talk, we'll be focusing on two different classifications. So those of you who are not aware of lipid vesicles, the two main types are LUVs, which are on the nanoscale, and GUVs, which are on the micron scale. And GUVs are particularly useful because they allow us to encapsulate large biomolecules, such as other vesicles or cells. And because they're cell size, we can also image them with fluorescence microscopy, or in microscopy in general. So we can monitor processes that occur inside, and we can also look at permeation and uptake, and also morphological changes. So the idea is that using GUVs as synthetic cells, we start with very simple membranes. We build up membrane complexity, and then multivesicular systems, and then high concentrations of proteins. And eventually, we integrate, various sets of machineries, such as membrane proteins, etc., to do more complex things, such as self-replication. So this is the kind of vision. So what do we do in this field? Well, we use engineering tools, so namely microfluidics, in two main areas. So we basically use them to create our compartments, and I will talk more about this later. And we also use them to capture our synthetic cells to then do manipulation and analysis. And within the field of bottom-up synthetic biology, we're interested in organized assemblies of tissues and synthetic cells. We're also interested in fusion and growth, multiple compartment systems, and finally, complex membranes. And as you've gathered from the title of this talk, I will be talking about the last two things for the majority of this talk. So, but before I start on how we use microfluidics, I want to start by talking about how we form them without microfluidics. So one of the easiest ways to form GVs is to use gentle hydration. So this is where you take a substrate, you dry your lipids, and you hydrate it with water, and you get the spontaneous formation of these giant vesicles. The advantages here is that it's simple. You can use physiological buffers, but it lacks the ability to encapsulate materials. The sizes are very polydisperse, and it's quite slow. So you can speed things up by applying electric field, but you still have the issue with polydispersity, low encapsulation, and also, in addition, if you apply an electric field, you sometimes have issues with salt buffers and things like that. The alternative method is the inverted emulsion technique, which is used by less people, but it's becoming more and more popular, whereby you take lipids in oil above an aqueous phase, and you incubate it and create a monolayer. Then you add in an emulsion at the top, which also has a monolayer around each emulsion, and then you centrifuge it through this interface, and those two monolayers zip together and form your GUVs. Here, the advantages are simplicity, it's quick, you have high encapsulation, but not very uniform. You can use salt buffers, and one of the big advantages here is that you can have asymmetry, both in the membranes and also the solutions. But the disadvantages are it's not always compatible with all lipids and proteins, and the sizes are still polydispersed. So, but having said that, we can still do lots of things with it. So one example here is that we can have communication between synthetic cells. So here we took advantage of the high encapsulation and encapsulated two populations of DNA, so 1 and 2 here, labeled red and green. We then mixed them together, and this was a collaboration with the Schwiller group in Munich. And then via freezing and thawing cycles, we could actually show that we had efficient exchange of DNA. So in this way, we had a kind of communication of genetic information between these model kind of protocells. Now, this is just a very quick example of how we can take advantage of high encapsulation using this technique. But what if we want a closer model of the membrane to actually how cellular biomembranes look like? Well, then we need to start thinking about membrane asymmetry, so to having different lipids on the inner and outer membrane. We also then want to start to include phase separation, because ultimately we want to have kind of complex membranes, or at least have the option to, and we want to have, with different proteins on different leaflets and in different positions. So this is one of the goals. Now, to do this, we can make asymmetric membranes using the inverted emulsion method. And I'm going to call it a double layer method because this is the way that it stands with most people using it. But the problem with this method is that actually, when you have one type of lipids on the emulsion and another type of lipids at the interface, you can actually get unwanted mixing of those two different lipids from the ones that aren't fully at the interface. So it's inconsistent for producing asymmetric membranes and not very reliable for producing phase-separated membranes as well. Having said that, you can still use it to make asymmetric membranes if you choose the right kind of material reliably. So for example, in this project here, what we were doing is we wanted to set up a bacterial membrane mimetic to study antimicrobial for antimicrobial studies. And in this case, we wanted to integrate lipopolysaccharides in one of the membranes. And here, the way that it worked quite nicely is that we could add the LPS from the water phase, not from the oil. So depending on the material that you want to integrate, that it can still work fairly reliably. However, we wanted to basically work on a new method, a new version of this that was more general and more reliable. So here I want to introduce a new method called the triple layer method. And the way it works is that you start by adding in your outer solution. In this case, it's a glucose solution. We then add in our lipids in oil. In this case, it's squalene. And the reason why we use squalene is because it has a lower affinity for cholesterol compared to mineral oil, which is normally used, and I'll explain why later. And then we have a longer incubation time to make sure we have a good monolayer formation. And then we add an additional spacer oil, which in this case is mineral oil, which is less dense, so it sits on top, and that doesn't contain lipids. And then we add our emulsion on top, which is mineral oil again. And this way we have kind of a separation of our two different lipids to reduce this kind of like unwanted mixing of the two different layers. And then when we centrifuge it through, we produce our GUVs. Now, how do they look like? Well, on the top line here, we just have an example of symmetric GUVs, just so show that it works just in a normal case. And on the bottom, we have asymmetric GUVs, where we have different labels, fluorescent labels, in the inner leaflet and the outer leaflet. Now, of course, you might look at that and say, well, what's the proof of that? It could just be in both leaflets, which is true. We can't visualize it. So what we do is we do an asymmetry test using a quenching assay with NBD labeled lipids and a quenching agent that we add. And the way that it works, for those of you who haven't done this before, is that in the case of symmetric GUVs, if we add the quencher to the outside, then we expect only the outer leaflet to be, the fluorescence to be quenched. So you would have half the signal. In the case of asymmetric, when the label is inside, there would be no exposure to the quencher. Sorry, when it was just inside, there would be no quenching here. So that's actually wrong. So you'd see no quenching. If it was asymmetric with a label outside, then you would expect complete quenching, so it goes to 0. And this is indeed what we see. So in the symmetric case, we have half the signal, and then in the asymmetric case, with the label outside, we have complete quenching. And there's just a movie showing you after we add the quenching agent, how it looks like. So of course, then we can actually quantify this for lots of different GUVs. And first, let's look at the traditional method, the double layer method, which was beforehand. So what we're looking for is around 50% quenching for the asymmetric case. And actually, we get about 60%. And we're looking for 0 in the asymmetric case here and 100% here in this asymmetric case. So it's not perfect. We have about 80% of the GUVs which are asymmetric. But we see an improvement with our triple layer method. So here we can see it goes close to 100% here and much lower in this case here. And when we compare the double layer to the triple layer method, we can see that about 93% of our GUVs are fully asymmetric compared to 80% previously reported. So we're quite happy with that. So we know the system works. So what can we do with it? Well, we wanted to have asymmetry with proteins. So what we did is we decorated the inside leaflet with nickel NTA lipids, and outside we added his tag GFP. And in this case, we saw no binding of the labeled proteins. So GFP is a green fluorescent protein at the membrane, which is right because there shouldn't be an exposure here. But then, and there's a movie of it happening here. But then when we flip it and have the nickel NTA on the outside, then we see binding of the GFP and enrichment at the membrane. So what this shows is basically that we now can have protein asymmetry as well as lipid asymmetry, and it supports the quenching data that we saw afterwards. So it really shows that we don't have any unwanted lipids in either leaflet. So we can have another example here where we added streptavidin inside one of our synthetic cells. And in this case, biotin, which you should bind to, is on the outside only. So we don't have any binding in this case, which is what you'd expect. And there's a movie just to show you there. But then when we add streptavidin also to the outside, we immediately see binding and enrichment at the outside. So this basically shows us the same thing as we saw beforehand, but in addition, this demonstrates that we can take advantage of the high encapsulation and put proteins both on the inside and the outside. So then we can combine this together and do two different proteins now. So in this case, we chose GM1 and cholorotoxin B on the outside, and on the inside we had biotin, related lipids, and streptavidin on the inside. And this is what we see. So we see enrichment of the streptavidin at the membrane on the inside, and then binding of cholorotoxin B on the outside. And then when we have the control, when we flip this, we can see that we don't have any binding around the wrong membranes. So that all makes sense. So we're happy about that. So what about phase separation? So we can make the membrane even more complex by now introducing phase separation. And for those of you who've tried to use kind of like the inverted emulsion method for phase separation, will know that it's not very reliable. And the main issue is that cholesterol, which you often need in the membrane to induce phase separation, is lost to the mineral oil phase. So we could get rid of the cholesterol and ignore it, but it's not particularly biomimetic. And we want a more general method that everybody could use. So this is not published yet, so it's under review. So hopefully soon you will be able to use this. And we could use DPPC to replace sphingomyelin, for example, but then we get mostly gel phase membranes and it's not biomimetic. So because we have squalene in our system rather than mineral oil, it has a lower affinity for cholesterol. So that means we get better reproducibility, and it's what we notice straight away. So we can reliably get phase separation with certain amounts of cholesterol, say from 20% up to 60% here. We also map this out on a phase diagram for those of you who are interested in this. And we vary the amount of cholesterol here to map out where we would get liquid ordered and liquid disordered phase separation. And when we compare this to a kind of oil-free method such as electroformation, which is kind of the gold standard method for making phase-separated membranes, we get pretty good agreement. So we're pretty happy with that. So what can we do with this? Well, what we can do is we can start to combine asymmetric membranes with phase separation. So in this case here, what we were doing is adding GM1, which binds to the liquid ordered domain, and then adding cholorotoxin B. And you can see that it enriches only in one of the domains here. So we get this kind of partitioning of our proteins. But this is not asymmetric yet. So we can introduce asymmetry by having GM1 only on the outside here. And then on the inside, we can have another degree of asymmetry by adding biotinylated lipids on the other phase on the inside. So we have streptavidin binding to the inside and cholotoxin B to the outside. So here we have two different proteins binding to different membrane leaflets, also at different lateral positions as well. So it's getting pretty complex. And one of the things that we noticed when we did this is we weren't expecting it, but we started to see these protrusions occurring, which you can see here. And here's just a kind of snapshots after we added the cholotoxin B, showing the development of these protrusions. And this wasn't just a one-off. We saw this quite consistently. Sorry, that's my kids in the background. And here we have other examples of where we saw these protrusions. There's also a movie happening here. And yeah, I won't go into too much details about this, but the reason why this happens, if you look in the literature, it's quite well known that when you have asymmetric distribution of large biomolecules on a membrane, it can change the spontaneous curvature of the membrane. And then just like cells do, it can result in membrane remodeling and new kind of morphologies and curvatures. So this is indeed what we're seeing in our system. So we thought, can we control this and get rid of it if we're not interested in having these protrusions? So we can to a certain degree. So what we can do is we can increase the amount of streptavidin on the inside, going from 10 nanomolar to 80 nanomolar here. And we can actually get rid of the protrusions in most cases. So we can decrease the membrane protrusion. So basically what's happening here is a kind of a tug of war between the different curvatures. So we can get a little bit of control over it. But we also thought, these are kind of static. So these are kind of like fixed things that we can then change. So this batch has this protrusion or this batch has another one. But can we make this more dynamic? And what we can do here is we can start to have osmolarity assisted budding. So in this way, we add glucose to the outside, which makes it hypertonic, which deflates the system. And that actually promotes budding of that smaller domain out of the kind of like the main one. So we can think of this as kind of a daughter and a mother vesicle, and a division event occurs. And here's just some snapshots here showing this happening over a few minutes. And if you also want to see, there's some movies of this occurring as well in a few examples here. So, we have this detachment and separation of these domains. And the interesting thing is that we only see this when we deflate normal phase-separated vesicles. We don't see this in our case. So we only see this when we have the asymmetric distribution of the proteins. So it really needs those proteins in specific places for this to happen. So this is a nice example of kind of like dynamic division of kind of protocells that we could use to divide synthetic cells. And it's something that we weren't expecting. So it's an emergent property that we couldn't predict it. So we were quite pleased with that. So this is just to summarize the first part of the talk where we talk about this. I've talked about this new triple layer method. And yeah, I've shown it to you. We can do the asymmetry, we can have dynamic division, and it's all very well and good. But there are still some issues with this technique. And these are that it's still kind of a bulk technique, so it's not really scalable or particularly high throughput, so you get a batch and that's it. The sizes of the vesicles that we get are still quite varied. There's not particularly very uniform encapsulation, so it can vary to a certain degree. And we still need to use densifying agents such as glucose and sucrose, so it's not particularly physiological. And as well as that, not all lipids and proteins can be incorporated. So this is why we sometimes resort to using microfluidics in our lab as well. So we use PDMS-based chips in our lab. And here you can see me holding one there. And we use these kind of flow focusing channels to produce water and oil droplets. And in the oil, we have our lipids. And we can do this obviously at quite high throughput rates, but these aren't GEVs yet. So what we do is we take that initial junction and we add another junction onto it. And that way we can create water in oil and water double emulsions. And then after extraction and removal of the oil, you're left with your GEV. So obviously we are not the pioneers of this, so there are other people who did this before us, but we take advantage of this because it can make monodispersed vesicles, which are tunable in size. So you can say, I want them to be exactly one size. It has really high encapsulation that's consistent, and it's continuous and high throughput. So it's scalable. So you can integrate this with other things if you want to. Now, we can use this in the topic of kind of bottom-up synthetic biology. You know, we've had some nice collaborations using this technique with the Tang group in Dresden, for example. And here we encapsulated coacevates, and via a pH change, we could initiate formation of these coacevates, these liquid droplets, and initiate enzymatic activity. So if you're interested, please look at those papers. But there are downsides to this, and this is mostly because in order to get these things working, you need to add in a lot of things. So you can see here on these diagrams, there's lots of things you need to add, such as ethanol, surfactants, polymers, glycerol, etc. And when we start to think of making more biomimetic synthetic cells, this is quite problematic, especially if we want to use things like enzymes and membrane proteins. So we wanted to see whether it was possible to create pure lipid vesicle membranes, but also have the advantages of microfluidics. And the spoiler alert is that we can. And this is our system, so it looks quite similar, but what you will see is that we don't have buffers, or we just have buffers and pure water and oil and lipids, and that's it. And we do this because we have higher lipid concentrations to stabilize our emulsions, higher flow rates, which we're able to form them quite nicely, and optimized channel dimensions, which I'll talk about later. So we can tune the flow rate ratios very, very carefully with our system. So we can start out with a thick oil layer, which you can kind of see illustrated here on these images, and we can tune it down so we can have a really, really thin oil layer, really, really thin double emulsion. And these movies here, which are slowed down, show you the before case where it's unoptimized and afterwards where it's optimized. And what you can see afterwards is they're hardly visible in most of the cases. So you can actually see, or actually you can't see them very well, which is exactly what you expect from having these thin double emulsion layers. So when we zoom out, this is kind of how it looks like with our production line. And just to remind you here, these are pure lipid kind of vesicles with just oil and lipids. And we can do this at quite high throughput rates, so around 3,000 per second. And because we have PDMS, it allows us then to have the option of kind of integrating this with other modules downstream if we want to in the future. So there's always the question of residual oil. So I will do one slide to convince you there's no residual oil in our system. We use octanol, which is not only biocompatible, but it's known to de-wet from the system. And you can see this happening here. So we have the kind of excess oil droplet, which begins to kind of like bud from the main kind of like GUV, which is being formed. But what we noticed is that even downstream, it wasn't completely detaching. And this was problematic because we really wanted to remove as much oil as possible. So we added this kind of serpentine module after the second junction. And the purpose of this is basically to slightly constrict the double emulsions as they pass, via the bend, and also to add additional shear stress to the system. And the shear stress basically helps to promote monolayer formation, and the constriction basically pops the oil layer off here, which you can see it's slightly removed here from the system. So in the end, when we collect them, we have GUVs and oil completely separated from each other. We also check for trace amounts of oil level by doing some diffusion measurements with FRAP to check to see whether we have the right kind of diffusion of the lipids, and we do. We also wanted to, we're quite thorough, so we also wanted to check for membrane functionability and also stability as well. So we take advantage of the other part of my lab, which is using microfluidic capture. So here we have a device, kind of a high throughput membrane analysis device, which allows us basically to capture about 100 GeVs in one of these kind of like buckets or nets. But we have over 200 of these per chip. So in the end, we can have 23,000 GeVs per device. We can still do fluidic exchanges. So we can add and remove things from the outside to do tests on our membranes. And in this case, what we were doing is we were adding alpha hemolysin pore just to make sure that we have a unilamellar membrane, there was no oil, and that we could functionalize them as well. We also took advantage of this and checked for leakage and stability, both using small molecules over time, dyes in this case, and also protein encapsulation. So everything was good with our microfluidic GeVs. They were kind of stable and leakage free. So how do they look like? Well, here's how they look like when we collect them. So there's lots of them. And when we zoom in, we see we have good monodispersity and good uniform encapsulation. And in this case, we were encapsulating labeled DNA plasmid. We can analyze each one of these GUVs and we can look at their sizes and we can see we have really good monodispersity. And we can also tune it as well. So in a single chip, we can change the flow rate ratios and we can change the size of our GUVs up to an order of magnitude. So down from about 10 microns to about 130. And we have really good uniform encapsulation, as you can see here. And just for reference with the traditional bulk emulsion method, it's much, much better. So it's versatile as well. So we can have a range of different lipids from neutral, charged, mixed of lipids, and a range of solutions as well from physiological buffers, growth media, even pure water if we want to. So in the context of bottom-up applications, it's really, really a versatile technique. And we can encapsulate a wide range of materials as well. So we can encapsulate DNA, as I mentioned, but also single cells, as well as non-biological components if you're interested in adding in, say, microparticles or something. So we have this nice way to make really nice, monodispersed GUVs. We've also shown that we can make complex membranes. So what can we do with them? So what I want to show you next is what we've done with developing our multiple compartment systems. So as you know, eukaryotic cells contain many membrane-bound organelles, and it's this kind of structural complexity which allows them to have, combined with the biochemical complexity, allows them to have a diverse range of functions. So if we want to build synthetic cells with, also a wide range of functions, I think it's essential we start to think about having multiple compartment systems. Not only this, but if we want to have them for applications in the future, then we can have, a wider range of applications such as drug delivery in this one example as well. So what do we want to do? So we would like to have signaling via reaction diffusion across multiple membrane compartments. More specifically, we want to have enzymatic reactions. So for example, we could just put 3 different enzymes inside one GUV and then having them all work together in a kind of in a cascade. And this wouldn't be particularly challenging for us, so we like a bit of a challenge in our lab. So we're going to set out some challenges here, and the first one is to have spatial control. So this is why we want to have multiple compartments, and we're going to use microfluidics to form this. We also want to have temporal control as well. So we want to be able to trigger our reaction whenever we want. So we don't just want to encapsulate them and then everything starts to work, then we have to struggle to find them and image them. We want to add them and then look at it. So this is why we have external chemical input delivered by microfluidics. And the last point is perhaps the most challenging point. We want to have directed transport. So we don't just want the intermediates in our enzyme cascade to be diffusing anywhere and everything all happening at once. We want to have a kind of directed pathway of our transport, so of our reaction. So for this, we want to integrate multiple membrane proteins, reconstitute them in our system. So what configuration to choose? So, we have a choice of different systems to work with. We could have multi-lamellar vesicles, which do technically have little compartments in there. We could use multisomes, we could use proto-tissues, but what we actually wanted to choose and what we finally decided to use were these MVVs or multivesicular vesicles. So this is just basically vesicles inside vesicles. Because it's the most flexible, so we can have different things inside each one, different membranes. It's a single defined container, which makes the analysis quite nice, and we can form them quite quickly as well. And about the formation, so how do we form them? Well, for those of you who work with microfluidics, you actually can use microfluidics to create multiple compartments. So you could have a kind of water in oil and water triple emulsion here. But the problem with this is it's difficult to get different things inside those different water compartments. And there's a lot of oil left over in such a system. So we would rather have a system like this, where we have a GUV on the outside and inside encapsulated using, for our kind of synthetic organelles, we're going to use the LUVs, which are on the nanoscale, which I mentioned at the beginning of the talk. And of course, we're going to use microfluidics to form them. So in this way, we add the preformed LUVs inside this channel and they end up inside the GUVs. And here's an image just showing you how they look like. So all the green dots here are individual LUVs encapsulated. And we still have good monodispersity in this system and good uniform encapsulation as well. So this is the kind of workflow of our system. So we use microfluidics to create our multi-compartment systems. We then capture them in another chip for high throughput analysis, and then we trigger them using microfluidic flow. And the idea is that after we add our input, we can then look at hundreds of different these micro reactors and read them out in real time, kind of at the single synthetic cell level. And the model kind of enzyme signaling network that we're using is quite straightforward here. So we have stachyose as our input, which reacts with alpha glucosidase to produce glucose. And then glucose reacts with glucose oxidase to produce hydrogen peroxide, which then catalyzes HRP to turn over amplex ultra-red and produce a fluorescent molecule. So in this case, what we're looking for is like a red fluorescent readout at the end of our enzymatic reaction. So let's start quite simple. So let's start with having everything all in one pot. So let's put all three enzymes inside one GUV. We need to make the membrane outside permeable by adding in alpha hemolysin. So this is a large pore protein which will allow stachyose to go through. And when we add that, after 10 minutes, we see an increase in the signal inside our GUVs. We can then quantify this for lots of different GUVs, and then we can see a nice plateau in the signal as the input molecule gets used up. We can perform controls without the input molecule and without the enzymes and show that only when we have these things present do we have a significant increase in the signal. So everything works. Let's make it a little bit more complicated. So let's add two compartments now. So here we have one population of LEVs inside, and we put the first two enzymes only inside those LEVs here. When we analyze it now, we can monitor both the LEVs inside and the GEVs to make sure everything is stable and not bursting, and the signal increase. And we have, again, a nice signal increase which plateaus over time, and we can perform the same controls as beforehand. Now, the final system to have this directed reaction pathway is a little bit more complicated. So now we have two kind of internal populations of synthetic organelles. And the idea here is that you can start to think about the input molecule when it meets the outer membrane. It's kind of like a low pass filter based on size. So normally it can't pass through. So the addition of the alpha hemolysin pore basically moves this kind of low pass filter to allow stachyose to come through. When it goes inside, it then has two options. So it can either go to one membrane here or this membrane here. Now the problem is that now we have this second low pass filter, which doesn't allow stachyose to go inside this other membrane pore protein, which is OMPF, because it's smaller. So it has to enter inside this compartment here. Then once it enters into that compartment, it's broken down by alpha glucosidase into a smaller glucose. It can then diffuse out and then is allowed basically to enter into this second compartment here. And then we complete the reaction. So you can start to think of this as kind of like, adding these things together with some kind of logic gate, in this case, an AND gate, if you like. And I want to point out one thing here is that there's an advantage here of having lipids as our choice of a material, because lipid membranes are kind of compatible with lots of obviously pre-existing membrane proteins. Now, of course, there's interest in using things like polymersomes, which are kind of semi-permeable, but in terms of reconstituting membrane proteins, there's limited things you can do with them at the moment. So here's what happened. So we wanted to encapsulate both of these on the chip because we didn't want to have pre-mixing of these two different populations. So we had to modify the microfluidic chips ever so slightly to have two inlets rather than one inlet, so that we could have on-chip mixing as shown here. So the two populations of LEVs get co-encapsulated and end up inside the GUV. And here we can actually label them separately to show that we have good co-encapsulation of our two populations. So here's how it looks like. So now we can monitor both LEV populations, the GUVs, and the signal increase. And then when we add the input molecule, we have a nice signal increase, which plateaus out over time. Here we have the same controls as beforehand, but in addition, here we have an additional control where we remove one of the pore proteins and show that we don't have any signal. So we need all these things working together in order for our synthetic cell to work. So you might have the question, does it depend on the size? So at the moment, they're all monodisperse. So we actually varied the size of our synthetic cells. And we actually noticed that for the smaller ones, the rate was lower and the amount of yield that we had at the end was also lower. Now, we hypothesize this is probably the effect of leakage, because what you've noticed is that we have these passive pores. Everything that can go in can also come out. So we did some tests using microfluidics, and indeed we saw that, for smaller GEVs, there was a pronounced effect of leakage, both into the vesicles and out as well, as you'd expect. So we understand why this happens, and what it actually highlights is it justifies the use of having microfluidics to create these monodispersed GEVs, because for each one of our GEVs, we have the same amount of leakage occurring. So it's kind of like, as long as we can understand what's happening, it's fine. So the fact that we have these controllable systems allows us to basically compare the level of complexities across these 4 different systems. So we can compare a bulk system without encapsulation to a 1, 2, and a 3 compartment system. And when we compare these all together, what we see is that we actually have a difference in the rate constants and the final kind of product yield. So the main difference you can see is that with encapsulation, with compartments, it's always higher than the bulk system. And this is most likely due to the smaller diffusers length scales. But one thing you'll notice is that the 1 compartment system always seems to work better than the 2 and the 3 compartment system. And this is most likely because it's quicker, because there's less barriers for the reaction to proceed, which makes sense. So you might also say at this point, well, Tom, what's the point in having multiple compartment systems? The 1 compartment system works better. Well, yeah, you might say that, but what we discovered is that when we did the single vesicle analysis of each one of these synthetic cells, we noticed that with the 3 compartment system, there was much less scatter in the data. Now, we don't know why this is fully yet, so we're working on this, but we know there's lots of different competing factors. So for example, in a multi-compartment system, the products can diffuse out into the next compartment, which shifts the equilibrium towards the enzyme substrate complex formation. We also maybe reduce any enzyme interference, which might occur in the 1 compartment system. But again, also, we don't have such a higher output because we have more barriers, more chance of leakage, etc. So it's a nice example that, even though we have half the intensity, it's still quite a lot. But it's an example of where we perhaps, if there was an application where we wanted to produce a molecule like a bioreactor, it's a more regulated system. So it's another advantage, well, example of an emergent property that we didn't predict when we set up this system. So that's quite nice. So this is the last slide. So yeah, we have this kind of like working synthetic eukaryotic cell system. And in the future, we could start to think about using this for drug delivery or bioreactors, as I mentioned, or even, sensors for responsive systems. But we could also use it to study biological things such as enzyme crosstalk and interference, the effect of crowding and diffusion, and also kind of communication as well. So, in the end, what we want to do is we want to start modeling this system to better understand, why we see what we do. But we also want to increase our complexity to bring about more functionalities. And the last thing I want to say is that life is complex. Let's try and make it simpler, because we might get more out of it. And with that, I will go to my acknowledgements and acknowledge people in my lab who have highlighted here, the ones who contributed to the work I showed today, and my collaborators over the years. And thanks for listening, and I'm happy to answer any questions.
Thank you so much. That was awesome. As a liposome person myself, I have to say I really appreciate this work. There's quite a bit of questions in chat. Can you see chat?
Oh, I'll have to exit from my shared screen, maybe. Can I see the chat without exiting from the shared screen? Oh yeah, here we go.
You
Yep.
should be, yeah. Could you read a
Um,
question out loud, because people watching the recording don't see the chat?
yeah. Great talk. Thank you. What is the size range of the protein that the membrane curvature system supports? How small, large does the protein need to be to produce the effect? Yeah, it's a good question. We didn't really look at the size and the molecular weight of the protein. I guess it will depend on it, but also maybe the density also of the decoration. So I think we mostly fixed the ligand, the kind of functionalized lipids we use, but I guess we could also play around with the size of the protein. That's right, and we could also play around with the density as well. So we could have more packing in there, but it's something we didn't look at directly, but I guess we could as well. Another one here. Once a GUV buds, can you get it to bud again? Or can a part of that bud off then undergo budding itself? There's nothing preventing that from happening. But with our system at the moment, the way that it worked was to have the different proteins decorated on different domains, and then these phase-separated domains then bud off. So we would have to add something else to the system. But in principle, yeah, we could get more budding off, but you'd have to add more materials, just like real cells. You can't have cells dividing forever. They have to also take up new material and input externally. But yeah, in the current system that we have, we can't do that, but we could think about that for the future. Yeah. Another one here. Can asymmetric lipid labeling in GEVs be used to track flip-flop in real time? Yeah, I guess so. Yeah. I mean, I don't know what kind of time scale, what real time that you were expecting to see, but I guess you could. Yeah, I can't think of a scenario where we could do that directly off the top of my head, but I guess if you wanted to set up an initially asymmetric, with something on the outside and then monitor over time if it goes to the other side, you could do that. With our ones at the moment, the ones that we have asymmetry with functionalized lipids and labeled lipids, and we're not expecting flip-flop with those ones. So normally, flip-flop happens with, cholesterol and things like that, but that happens very, very quickly. Maybe with labeled cholesterol, I don't know. But yeah, in principle you could, but not with the ones we have. And then Sarah Keller replied to that as well. I won't read that out. Thanks, Sarah. Very interesting work. In the multi-compartment system, what did you try in terms of different types of lipids, increasing permeability of membrane to help with efficiencies? Yeah, we didn't change the type of lipids in our multi-compartment system because, the system as it was, I will go back to that final slide there. It to us, and for those of you working with GVs, this is quite complicated. For those of you who are cell biologists, this is not complicated at all. It's very simple, but for us, it's quite complicated. So we didn't want to mess around with the system too much. So it was working, and we were very happy with that. It was reproducible. So we didn't explore different types of lipids and playing around with the permeability. But like I said at the end, this system is really tunable. So we could change a lot of things. We could play around with the lipids to change the permeability. We could, more to the point, we could change the density of the membrane pore proteins that we work with. So there's lots of different variables, and it's quite exciting because you could change the concentration of one population, you could change the size of everything. So, we set up our system as it is, and this is obviously published. You can read about this. Because it gave us a nice time scale to work with. Everything was working in 10 minutes. So, it gave us a nice time scale to work with. But in principle, we could tune it to go slower, faster, more or less efficient. Yeah. When you have nested liposomes, what happens when you extrude them? Okay. What happens to the internal liposome when the external one gets divided? Okay. The first question is, I don't know. Normally, when you extrude GUVs, you can make them smaller. So I guess it would just burst the GUVs. I don't know. It would be a disaster for them, I guess. But if you wanted to make them more robust, then I don't know, maybe that would be a good test. Second question is, what happens to the internal liposome while the external one gets divided during extrusion? Yeah, I don't know. I can only speculate. I'm not sure. Question from Sarah Keller. How do you add alpha hemolysin and OMPF? Are they simply in the buffer solution and they self-insert previously formed? Yeah, good question. So for the alpha hemolysin, you add it to the solution afterwards, so it self-assembles. So it's a monomer, and it readily self-assembles in the membrane, and we did that because it's really simple and really easy to use. For the OMPF, you need to use membrane reconstitution, so detergent-assisted integration and then biobeads, so all that stuff. But we prepare our populations of LEVs kind of externally beforehand, so you can make normal LEVs as you like with protein reconstitution and then encapsulate them later. Yeah. Um,
Okay.
the last question there. Um, oh no,
Um,
one more.
yeah, you got one more.
Okay, yeah, it's good. We have time, right? Yeah. Do you have an outlook towards building a gene expression into your systems or collaborating with others to facilitate that? Yes, yes. It's one of the things we want to do and we're looking into at the moment. So nothing to report at the moment, but yeah. Um, Sarah asked a follow-up question. Is the OMPF channels always open? My memory is that Alpha Immunolysis is always open. Yes, they're both always open, so they're passive channels. I don't know if you can close them with some chemicals, I think. There's possibility to block them, but they're not passive channels. They can't be open and closed, as far as I'm aware, and certainly not in our case. So yes, this is a disadvantage of our system. It's a diffusive based system in a sense talk, it's a disadvantage. So everything that our vesicles produce can also then leak out again. But on the time scale that we were working is that wasn't such a big issue. But yeah, we're interested in making, better channels, integrating better channels in the future, because that would be really cool to have kind of more directed channels, more active channels. But not yet.
I second that. If anyone can make better channels, there's a lot of uses for that.
Yeah, exactly. Then you could really start to make more efficient systems. You can direct things to certain compartments that you want to. Yeah, it's something that is it's going to be more complicated, but yeah,
Mhm.
if we can do it, then it opens up a lot of possibilities. Yeah, it's quite exciting.
Something, because right now we have the really complex stuff, like we have ATP synthetase sex system style stuff, and then on the other end we pretty much have alpha chemolysin,
Hmm.
which is awesome. I love that protein, but it's the dumbest channel you've ever seen.
Yeah.
And we need stuff in the middle. We need more stuff in the middle.
Yeah, yeah. I mean, yeah, for our application, it was nice to use it because it was, it has different size. You can get these different
Mhm.
sizes, and we use that size selectivity to our advantage in this case. But yeah, for moving on, it has limited applications as well. Yeah, we
Mhm.
basically need to start, people working on these bottom-up approaches, you need to start collaborating with labs which, produce these pores and reconstitute them and get them into our bottom-up systems. This is the way to go. Yeah.
Mhm. Thank you so much. I think if there are no more questions, um, I was really intrigued by that question about the extrusion. I would love to see what happens to the
Yeah, I'm not
inside
quite sure.
ones. But
Maybe the person could add a follow-up comment if they're still there. Why they would want to extrude them. I wouldn't want to extrude GEVs that I've just created
yeah.
like this because it would most likely destroy them. But maybe what I mentioned, maybe there's a need to see how robust they are, because if you wanted to use them, in biomedical medical applications, then maybe you
Mhm.
need to show that they're more robust. Maybe
Mhm.
extrusion would divide them and you would get two populations, but they would have the same
Yeah.
things in either one, most likely, because these diagrams here are a little bit misleading, right? So
Yeah.
they're not just two inside. There's a
Mhm.
population, equal amounts of concentration, with thousands of these LEVs inside, in equal amounts. So they're
Mhm.
equally distributed throughout the system. So extruding them and popping them and dividing them mechanically like that would probably result in just a smaller version of it. But then you would also get bursting and leakage and things like this, and probably
Mhm.
less concentration in the end. Yeah.
Yeah. All right. Thanks so much. Honestly, I would just want to extrude them for the heck of it because I'm curious what would happen. Uh,
Yeah. To see if they still work, you mean?
yeah,
Yeah.
exactly.
Yeah. Yeah.
Uh,
I didn't want to. They're my little babies.
yeah. They would be even littler if you extrude them. You would have more of
Yeah.
them.
Yeah, but the problem then is like if they're smaller, they're harder to analyze.
Oh
At the
yeah.
moment, at the moment, I think we were on average around 60 microns. So they're nice, bigger than cells, nice big vesicles that we could easily image over time and get good data from it.
Yes,
Smaller you are,
totally,
then less signal to noise. It's a problem, but yeah, not impossible.
totally.
Cool.
Great. Thank you so much, Tom. And thanks everyone else for a fantastic discussion.
Yeah, thanks everyone.
And have a great rest of your day.
Yeah, you too.
Bye.
Okay, bye-bye.